Hello guys, welcome to my channel. So if you saw the previous video, we are talking about the protein structure and function. So this was that we talked in the previous video. So let's just do a quick review before we go further into the topic. So as we know, proteins are the main building blocks of amino acids. So there are different kinds. Of proteins, such as like enzyme, structure protein, motor protein, signal protein, storage proteins, receptor proteins. So there's just different kinds of proteins. And so in order to study the proteins, so the proteins are the polymer of amino acids that is here. And so that's why we need to understand amino acids, right? So there are like 20 amino acids in particular. From there, all the proteins, all different kinds of protein, that is enzyme, structure, storage proteins, specialized protein, all are made from this 20 amino acids, right? So the first that was discovered was asparagine, right? That is in 1806, and that found in asparagus, right? So the asparagine was found in asparagus, and the last was 309. That was last discovered, and the name of the amino acids are just as simple as that. That is found from asparagus. That's why it's asparagine. Same glutamate. That's from wheat gluten. That's why tyrosine derived from cheese. The Greek name of cheese was tyros. So that's why. And to understand the amino acid, like there are 20 amino acids, so like we divided that amino acids into five main classes of amino acids that are non-polar and aliphatic R group, aromatic R group, polar uncharged R group, and positively charged R group, and negatively charged acidic group as well. All right, so these are the five classes of amino acids, and all that 20 are divided in this five classes. All right, and this amino acid names are done in the abbreviation of three-letter abbreviation or three-letter code, and with one symbol. So for example, alanine is ALA, the abbreviation, and the symbol is A. So that's so on for this, all these 20 names. And the amino acid has the structure of, that is, it has a one alpha carbon atom like this in the middle, and then it has an amino group, it has a carboxylic group, it has a hydrogen atom, and it has a side chain R. And this all proteins are all defined from this side chain or differentiate from this side chain. And except everything, the glycine has hydrogen atom instead of the side chain. All right, so this was the topic from from the previous video. So now we'll see further and study about the amino acids. All right, so now we'll study about the uncommon amino acids. So there are uncommon as well. So the common amino acids are this 20 amino acid. From there the proteins are built, but there are uncommon amino acids as well, and their functions are also very important. Uncommon amino acids. All right. Right. So what are the uncommon amino acids? So they are also known as the modified amino acids. Why they are known as modified amino acids? Because when there is a synthesis of a protein that has been completed, a few amino acid residues. So what is the few amino acid residue? So like suppose when there are two amino acids are joining together, they will eliminate a water element. All right, and there are other residues as well. So a few amino acid residue in the primary sequence may further be modified. So when the primary sequence of amino acids are joining, right, and the proteins are about to create, so there the residues from the primary sequence that can be modified in an enzyme catalyzed reaction that may add a chemical group to it or oxidize or otherwise modify specific amino acid in the protein. So because protein synthesis occur by a process known as translation, we'll study that translation later, but just for this knowledge right now. And these changes that are to the amino acids residue are called post-translation modification. So there are more than 100 different post-translation modified amino acids residue have been found in human proteins. Right? So some of the uncommon amino acids are, so the one is 4, number it 1 here, 4 hydroxyproline. All right, so this 4 hydroxyproline is a derivative of proline. All right. Derivative of proline, right? And same, there is like 5 hydroxy lysine, and that's a derivative of, all right. So this are two uncommon or modified amino acid, and this one is found in plant protein wall. This one is found in plant protein cell wall, all cell wall protein, and this both are found in collagen, right? A fibrous, what is a collagen? That is a fibrous protein of connective tissues, right? There is another, so that is 6 and methyl lysine. So that is a constituent of myosin, right? That's a contractile protein. The myosin is, what's myosin? That's a contractile protein of muscles, right? There is another important and common as amino acid, that is gamma carboxyglutamate. Gamma carboxyglutamate. So this is one. That's another important protein. This protein is found in the blood clotting protein. This is found in, this amino acid is found in blood clotting protein, prothrombin. All right, and there is certain other calcium channels as well. There is a part of biological function too. And there is a more complex one, that is desmosin. That's a derivative of 4 lyse residues. So that's one something complicated. But let's just go through the name of that. Desmosin. That's a derivative of lyse residue. All right, and this is found in protein, fibrous protein elastin. Found in elastin. All right. So these are some of the uncommon or modified amino acid. There are some as well, like special cases, selenocysteine or pyrrolysine. Some special cases. Pyrrolysine. All right, these are the rare amino acid. Why they are the rare amino acid? Because selenocysteine contains selenium rather than sulfur of cysteine. Because cysteine has sulfur. Like if you see the picture, like that's the structure of cysteine and it has sulfur. But instead of sulfur, this contain selenium that is actually derived from serine. Selenocysteine is constituted of just a few known protein. So there are like some 300 additional, so these are just a few, but like there are like 300 additional amino acids that have been found in cells, right? And they have a variety of functions, but not all are constituents of proteins. Remember that. Like special note to ornithine and citrulline. Ornithine, I will just write it down here. Ornithine. Citrulline. Why these are important? Because they are key intermediate metabolites in the biosynthesis of arginine and in the urea cycle. We'll study later in when we'll go through the different metabolic pathways. So these two are important in that biosynthesis. All right. All right, so this was uncommon amino acids, and so there is one more function of amino acid. Like amino acid can act as an acid and base as well. Amino acid can act as an acid and a base. Bases, sorry. All right. So why they can act as an acid and bases at the same time? Because the amino and the carboxylic group of amino acids. So if you remember, it has a carboxylic group, it has amino group, right? It has R and the hydrogen atom, right? So because of the carboxylic and the amino group, along with the ionizable R group of some of the amino acid function as a weak acid and weak bases, right? Because when an atom is lacking this, if the atom does not have an ionizable R group that is dissolved in water at the neutral pH, that is 7, it exists as a dipolar ion, or also it's known as Victorian. Ion that is known as hybrid ion, which can act either as an acid or base. So remember, the substance that has dual property, dual nature, that can either act as an acid or base are known as amphoteric. All right, they are known as amphoteric, or also often known as ampholites. So when you hear the term ampholites, that means this element can be either as it has both either as an acid or can react as a base as well. All right, and that's as simple as that, because it has the carboxylic group and the amino group, so it can take a hydrogen ion and make it COOH, a base, or it can give it and make it as an acid. All right, so now we saw that common amino acid and the uncommon amino acid. So now we'll move further and study peptides and proteins. Peptides and proteins. Right, so two amino acid molecule can be covalently joined through a substituted amide linkage termed as a peptide bond to yield a dipeptide. So if two amino acids join together, it makes a dipeptide, and such a linkage is formed by a removal of element of water, that is by dehydration, from the alpha carboxylic group of one amino acid and alpha amino group of another amino acid. All right, so I will show you the reaction. So for example, so this is the first side chain. Oops. This. Okay, R1. So let's just suppose this is R1. This is, all right. This is the first. So this is the carboxylic group. This is with the hydrogen atom. All right, and this is the second one. And this has again the CH. This is R2. That's the second side chain with this. All right. So when this two reacts together, so there will be elimination of H2O, and how they will join. This is R1, right? So this C, that's double, that's from this carboxylic, that will be joined by this here. You can see that. Okay, you can see that. This is R2, that's the second one. C double O. So this part. So that's a peptide bond between two amino acid. All right. So the first amino acid and the second amino acid joined together by the condensation reaction. All right. So they are joined by a condensation reaction to form dipeptide, right? And what's the condensation reaction? Water is expelled from that. So H2O. So H, that's one H from the amino, one hydrogen atom from the amino group, one hydrogen atom from the carboxylic group. That's gone with the oxygen from the carboxylic group. So that has, this is joined with this. All right, and that's the dipeptide bond. This is just a simple example of a reaction that is shown in the equilibrium, but the actual, to make the reaction thermodynamically more favorable, the carboxylic group must be chemically modified or activated so that hydroxyl group can more readily eliminate it. All right, so the peptide bond formation is an example of condensation reaction, right? That's a common class of reaction that occurs in the living cell. All right, so when two amino acids join together, that form dipeptide. Right, you can see that, right? Dipeptide. When three amino acids join by two peptide bonds, like this is one peptide bond. When three amino acids join by two peptide bond, it is called tripeptide. Similarly, four amino acid can be linked to form tetrapeptide, five for pentapeptide. So a few of the amino acids, more than 5, 6, 7, 8, that is called oligopeptide. And when many hundreds of amino acids join, that product is called polypeptide, correct? So protein may have thousands of amino acid residues. So although the term the protein and the polypeptide are sometimes used interchangeably, but the difference is the polypeptide generally have a molecular weight of below 10,000, but the protein have a higher molecular weight. And uh, just when an amino acid sequence or a peptide or a polypeptide or protein is displayed like this, so the amino terminal end, so this is an amino terminal end, all right, is placed on the left, always placed on the left, and the carboxylic terminal on the right, and the sequence that is read from left to right. So like for example, this is alanine and this is glycosine, so like it will be AG like this. So it will go from left to right, beginning from the amino terminal end. As this is a condensation reaction, so the hydrolysis of peptide bond is not that quite easy. It occurs only slowly because it has a high activation energy. As a result, the peptide bond in proteins are quite stable with an average half-life of about 7 years under the most intracellular conditions. So the peptide bond half-life is, or the protein half-life is 7 years. Right, so this is how the protein is formed by joining two amino acids, and some protein contain chemical groups other than amino acids as well. So like it's not always all proteins have this amino group, but some proteins are that they have other chemical groups as well. So for example, the enzyme ribonuclease A and the chymotrypsin contains only amino acid residues and no other chemical constituents. So it does not contain the actual amino acid, but it contains an amino acid residue and no other chemical constituent. These are considered as a simple protein, but there are some proteins contain permanently associated chemical component in addition to the amino acids. So there are amino acids, but beside the amino acid, there are some other chemical component as well. So that type of protein is called conjugated protein. All right. So I'll write it here: proteins that contains chemical component with amino acid, you can see that, right, are termed as conjugated proteins. Right, they are termed as a, or called as a conjugated protein. So the non-amino acid part of the conjugated protein is usually called its prosthetic group, right? So like whatever group for are with this amino acid, so it will be called a prosthetic group, right? That's an outside group. Or conjugated proteins are classified on the basis of the chemical nature of their prosthetic group. So like the conjugate proteins are classified on the base, like which prosthetic group it contains. For example, lipoprotein contains lipid, right? So the example is lipoproteins contains lipids, right? Glycoproteins contain sugar group, metalloproteins contain a specific metal group, right? So on your screen, you will see a few of the conjugated protein and their examples as well, right? So phosphoproteins, hemoproteins, right? These are called as the conjugated proteins because it has, beside the amino acid group, it has some other chemical component. So to understand the protein, so for the biochemist to understand the protein structure and function, so how they studied the protein. So to study a protein in a detail, what the researchers do, like they must be able to separate it from other proteins in a pure form and must have a techniques to determine its property. Right. So this is just for a knowledge, like how what techniques they used. Like I will not go very deep in. I will just go through the names, what technique they use usually to separate protein. So pure preparation is essential before the protein properties and activities can be determined. So the first step that in protein purification procedure is to break open the cells and releasing their proteins into a solution that is called a crude extract. Once the extract or organelle preparation is ready, various methods are available for purifying one or more of the protein it contains. The most common one, the extract is subjected to treatment that separate the proteins into different fractions based on the property such as size or charge, right? That how it differentiate based on the size and charge, and that is referred to as a fractionation, right? That process is known as fractionation. So early fractionation steps in purification utilizes difference in protein solubility, which is a complex function of pH, temperature, salt concentration, and other factors. So the most powerful method of fractionating protein make use of column chromatography. That's one of them, which take advantages of difference in the protein charge, size, binding affinity, and other properties as well. There is ion exchange chromatography as well that exploits differences in the sign and the magnitude of the net electric charge of proteins at a given pH. So there is like affinity chromatography as well. And like for this fractionating, the solution containing protein of interest usually must be further altered before the subsequent purification steps are possible. For example, you can see like that is a dialysis is a procedure that separates proteins from small solutes by taking advantage of proteins larger size. So what they do, the partially purified extract is first placed in a bag or tube made up of semi-permeable membrane, and when this is suspended in much larger volume of buffered solution of appropriate ionic strength, what the membrane will do, the membrane will allow the exchange of salt and buffer but not the proteins. So thus dialysis retain the large protein within the membranous bag or tube, allowing the concentration of other solutes in the protein preparation to change until they come into an equilibrium with the solution outside the membrane. So dialysis might be used, for example, to remove an ammonium sulfate from the protein preparation. And there is one more technique as well. Protein can be separated and characterized by electrophoresis. So this is an important technique for the separation of protein, is based on the migration of charged protein in an electric field, that is called as electrophoresis. That process is called as electrophoresis, right? This procedure is not generally used to purify protein because simpler alternatives are usually available, and electrophoretic methods often adversely affect the structure and thus the functions of protein, right? So that was just a few of the techniques or procedure for purification of protein, and that purification of protein is actually usually just only a prelude to a detailed biochemical dissection of its structure and function. So the question arises, like what is that makes one protein an enzyme, another a hormone, and another a structure protein? So, right, and like another is antibody, how they differ chemically. So the most obvious distinction are structural. So that's why we'll study the structure of protein, right? So now we'll study the structure of protein. All right, so I will just remove this. All right, so What we'll study now? Structure of protein. Alright, so let me just do this protein structure, and that is now we'll study primary structure of protein.